Let's go ahead and get started. I do want to thank everybody again for joining us today for day 9 of 9 for our 2020 Patient Educational Conference. Hopefully everyone's been able to view the videos that we've had available the 8 days prior to this, and we'll have this is being recorded just so everyone knows. So it looks like nobody has like images or photos of themselves up, but just FYI, if you do, it might be visible in the eventual recording that we are putting later today on our YouTube channel and on the event page. So just kind of be aware of that. I've also muted everybody, and I'll mute myself here in just a moment as well. And we will have everyone just stay muted during the presentation, just so any background noise that we might have going on in our homes doesn't take away from what Dr. Sachdev is sharing for us today. And then at the completion of the presentation, Dr. Sachdev will take any questions at that time, and then we can go ahead and unmute for those questions, or feel free to type them in the chat box, and I will keep an eye on that, and we'll make sure that we address those at the end of the talk as well. So yeah, let's get going. Thank you, Dr. Sachdev.
Of course. So we are confronting today the topic of clinical trials update in myasthenia gravis. And again, I'm Amit Sachdev. My hats over at Michigan State University include being the Associate Chief Medical Officer for MSU Healthcare, the Medical Director for Neurology, and I am the MDA Clinic Director. I'm actively involved in clinical trials in myasthenia gravis. We have 6 of them in various stages, either closing, enrolling, opening, etc. And I care for approximately 150 patients annually with myasthenia gravis and across the subtype of MG, acetylcholine receptor positive, MuSK, Lambert-Eaton, you name it. And so I'm very pleased and honored to join the Myasthenia Gravis Foundation for the annual meeting. They've had the great pleasure of speaking at this meeting for the last 5 years. And every time I get to give this talk, I think it's a little bit more complex, which is exciting. There we go. I do have some disclosures. As you can see, we have some clinical trial partners. I am also on some speakers bureaus. I get funding from various nonprofit agencies, and I sit on various boards, including sitting on the board of MGMI. I have provided scientific advisory opinions for a number of companies, and then I have a boatload of student loan debt, and that is held by the U.S. Department of Education. And I'd like to include that one in the disclosure because it definitely keeps me on the good side of the U.S. federal government. Additional disclosures. When I joined MSU, of course, I had more hair. Now I have less. The MSU has something to do with that, but my children do as well. And then I can't function without my team. And so this is the varied members of the team. There are so many of them. And from not just MSU, but from some of our third-party partners as well. And then the final disclosure is that we are going to talk about some off-label and experimental use of medications. With the exception of eculizumab, every medication that we talk about is either going to be experimental or off-label. So for a quick roadmap, we're going to talk about an introduction to myasthenia pathology. Given the arc of this conference, we're not going to spend a lot of time there. We're then going to talk about the role for things like T cells, B cells, about interfering with antibodies as they get to the muscle, and then interfering with what they do at the muscle. And these are the various treatment strategies that we have for the management of myasthenia gravis. And we're going to try to talk through the strategies in this way because the experimental medications that are being deployed for myasthenia gravis are deployed targeting at these various levels of management. All right, so for the basics, over here you see a muscle, and on that muscle you see a receptor. Over here you have a nerve, and a nerve releases a packet of chemical. That chemical is acetylcholine. So the nerve releases the acetylcholine drifts over to the receptor, and it makes the receptor, it makes the muscle do something and twitch. Antibodies get introduced into this scenario. Antibodies are produced by the immune system, And what they do is they interfere with the receptor. So that's the basics for how myasthenia gravis works. What we're now going to do is we're going to introduce how clinical trials work. So clinical trials have 4 phases. The first phase is often a safety phase, and it involves healthy volunteers. It defines whether or not you can safely give something to people. The second phase is often dose finding and endpoint finding. So what does that mean? It means we don't know what the right dose is for a various study agent, and we're not exactly sure the ways in which it changes the disease. So we're going to try to figure out what's the best way to measure the disease. Is it safe for people who have the disease? And if you give people various doses, in what way does it modify their disease experience? Phase 3 is the most important phase. These are often large studies. They are often a single dose versus a placebo. A placebo is sugar water, an inert substance, some substance that does not do anything. These are often double-blind. Double-blind means that neither the patient nor the doctor knows what the patient is getting. And often they include an extension study. Now, that an extension study is really important. The reason that it's important is because during the placebo-controlled phase of the study, you will not know whether or not you are getting the drug. But during the extension phase of the study, you are guaranteed access to the drug. You know you're getting it. And so the reason many patients enroll in clinical trials is because there is some added benefit, we think, from these experimental medications. It might be a good choice for a certain subgroup. And many patients are willing to go through the placebo-controlled phase because they will gain access in the guaranteed access phase, the extension study. Phase 4 is the post-marketing. So once a drug is approved, it has to then be watched. And so phase 4 is post-marketing, watching the drug after it's been approved, but also trying to study it in certain interesting subgroups. A lot of pregnancy data is achieved in phase 4. So when we think through therapies in myasthenia gravis, we're going to think about T cells, B cells, antibodies path towards the disease, towards the muscle, and then its effect at the muscle. And so let's first talk about T cells. So T cells come in a couple varieties: regulators, killers, and helpers. Regulators, killers, and helpers have different functions within the immune system, but you can roughly think of these as master switches. They will guide the activity level of the immune system. And so that's the role of the T cell. There are many oral drugs that we use off-label to treat myasthenia gravis that act on the T cells. The oral drugs are thiopurine, the trade name there being Imuran, cyclosporin, mycophenolate, the trade name being Cellcept. So let's go through a diagram here. Again, you have nerve and you have muscle. You have acetylcholine, which transmits from the nerve over to the muscle. You have the presence of the T cells, regulators, helpers, and killers. And these guys influence what is going on in terms of attack at this junction. These drugs, as thiopurine, cyclosporin, and mycophenolate, block or regulate activity of the T cells. Now you'll see here that there's this long string of letters and numbers and dashes. This is an experimental agent. So as you can imagine, if we think of the T cells as regulators or master switches, it might make sense that if we could influence their activity and turn their activity back to normal, that we might be able to slow down the aggressiveness of immune responses or restore normal balance in the system. Might be one way of thinking of it. So this class of agents is a class of agents that helps to change the tolerance of the system. And these are very early phase agents. These tolerance agents are stage 1, just deciding whether or not they are safe. This is the newest class of agents that we will be talking about for myasthenia management and in general, more in general in autoimmune disease management. And this class of agents will probably start to emerge into trials in about 4 or 5 years. Now, the second thing, your group of cells that are involved in immune responses are the B cells. So the T cells can be thought of as master switches. The B cells can be thought of as the factories that actually produce antibodies. So what is the role of the plasma or the B cells? They produce antibodies. Antibodies attach to targets, and when they attach to targets, they cause disease to happen. So in myasthenia, antibodies cross-link the receptors. They cause those receptors to be degraded, and they induce inflammation. And so it stands to reason that if you could eliminate these plasma or B cells, you could significantly drop the production of the antibodies. And if you could drop the production of the antibodies, then you could interfere with the disease process. So what we have here is a graph of antibody level over time. And what you can envision is that there is a dose right here of a drug that might interfere with antibody production or that might be toxic to plasma or B cells. And so if you introduce a drug right here that is toxic to plasma or B cells, then you will reduce your antibody production. And that is exactly what the drug on the market, Rituxan, does. Rituxan targets CD20 cells. There is an experimental medication called TAK-079. TAK-079 targets CD38 cells, and that is a trial that is actively enrolling at this time. And so antibody levels would reduce if you eliminate this class of cells, plasma cells or B cells. And there is at least one agent currently FDA approved, but not approved for myasthenia gravis, and another agent that is in clinical trial that does just this. We are a site for the TAK-079 trial. So what does this look like in the picture diagram? All right, so T cells, recall, they are the master switches. They regulate the activity of the immune system. They also have specific functions in the immune system where they can go and directly damage things. And we have a group of drugs that regulates their activity. And we have a group of experimental drugs that attempts to restore normal activity. Now, these T cells influence the function of B or plasma cells. B or plasma cells produce antibodies. These antibodies get introduced into the bloodstream. So what you're looking at here is a blood vessel. So the antibodies are in the blood vessel, and then they are going to go and attach to the receptor on the muscle. Rituxan and Takeda's new agent, these two drugs interfere with B cells and plasma cells. And the result is, once you eliminate these antibodies, you have a drop in, eliminate the production of these antibodies, you have a drop in antibody levels in the bloodstream. And so you eliminate the B cells, therefore you eliminate the antibodies. All right, so what if you could prevent the antibodies from getting to the muscle in the first place? And so that was an interesting idea because it doesn't sound like it's a good strategy to eliminate T cells or eliminate B cells. As you can imagine, you need B cells and you need T cells in order to mount good immune responses. And so in particular, in the setting of a pandemic, it is very logical that we would be concerned about eliminating T or B cells or interfering with their function. And so what we might instead want to do is we might want to interfere with the way that antibodies get to the muscle, or we might want to interfere with what they get to do when they attach to the muscle. And so that's exactly what plasma exchange does. Plasma exchange depletes antibodies from your bloodstream. So you insert catheters, pull the blood out and filter it. IVIG replaces antibodies. And then an experimental class of drugs, the FcRn inhibitors, deplete antibodies by breaking them down. So plasma exchange depletes antibodies by drawing them out. FcRn inhibitors depletes antibodies by breaking them down, and IVIG replaces antibodies. And so what you see here on this graph is the drop in antibody level if you were to use plasma exchange or FcRn inhibitors. And this graph looks a lot like the graph that you would have seen for rituximab or for the new Takeda experimental agent. And what you see is a precipitous drop in antibody production. And so the graph looks similar, but the key focus here is that the infrastructure that produces antibodies is still intact. What that means is that this graph actually recovers in weeks rather than in months with the depleting drugs. Over here, this is IVIG. With IVIG, in fact, what you do is you very quickly ramp up antibodies in the bloodstream. You flood somebody with antibodies, and then they very quickly break down. And so what this is this is the level of antibodies that the person that has produced on their own. As you're flooding the person with antibodies, they are breaking down some of their own, and then there's a slow trend back up. So you deplete antibodies with a plasma exchange or FcRn inhibitors, and this is what your graph looks like. So again, let's walk through it. You have T cells, which either regulate or go and kill. And you can modulate the activity of T cells with various either on-market but not FDA-approved drugs or various experimental drugs that are on their way. You've got B cells. B cells are the factories. They produce antibodies. You can draw those antibodies out. This is a rough way of trying to demonstrate a syringe. So you create a different path, right? You introduce a syringe, the antibodies follow that path. You introduce a filter, that's what this little blue line is. The blood goes back in, antibodies are gone. They never get to the muscle. That's what plasma exchange looks like. Or, oops, you can degrade the antibodies. So this is the experimental method, the FcRn inhibitors. So let's talk about this topic for a little bit. The lining of the blood vessel, recall this is a blood vessel right here, and that's the path that the antibody takes from the B cell to the muscle. The lining of the blood vessel is a living tissue. The blood vessel is not a pipe. It participates in managing the health and consistency of the blood that flows through it. It is an organ. And so that is a remarkable way to think about blood vessels. We never thought of it that way before. But in fact, blood vessels are organs. They are not just pipes. And one natural thing that blood vessels do is they participate in metabolizing or cleaning the blood that runs through them. And so the way that blood vessels do that is they take in some of the materials from the bloodstream, and they will just do this randomly. When they take that blood in, the antibodies will be a part of what the blood lining, the lining of the blood vessels just takes in on its own. Now, the blood vessel can choose to destroy the antibody, or it can choose to spit it back out. And so if you block the path where it spits it back out, it will go and destroy the antibody. So we're going to talk a little bit more about this. The FcRn inhibitors is a very busy area of development. There are 4 companies involved: argenx, UCB Pharma, which recently entered the space not just in FcRn inhibitors, but also complement inhibitors; Momenta Pharmaceuticals, and Immunovant. And so these 4 companies are all developing FcRn inhibitors, and they are all running clinical trials in myasthenia gravis. The Momenta trial is a trial we actually have hosted the phase 2 at Michigan State University. That phase 2 is now concluding. We will learn more as time goes on about the phase 3 and whether or not that will be available at MSU as well. Argenx is a clinical trial partner at Michigan State University as well. We are hosting a phase 3 trial with this drug, efgartigimod. So these 4 companies are developing these FcRn inhibitors. And again, what does the FcRn inhibitor do? It keeps the antibody from being preserved in the bloodstream. So one way that you metabolize is the lining of the bloodstream may take it in and destroy it. And the FcRn inhibitor promotes that avenue. Okay. So replace antibodies. This is what IVIG does, right? The black here is your total antibodies. The red here is the antibodies you produced on your own. With IVIG, we very quickly flood you with antibodies. You cannot possibly hold all of those antibodies. And because you couldn't possibly hold all of them, you couldn't recycle all of them, the antibodies that are running through the blood vessels and are taken into the lining get destroyed. And so you're flooded with many more antibodies than you could ever hold. Your own antibody levels plummet, but that's okay because you've got the antibodies you were flooded with from donors. And so you get to keep the immune profile of everyone else. So if most people have been exposed to, for example, the flu from 2019, then you are still carrying antibodies against the 2019 flu. Okay, so what does this look like? Here is our diagram again. So here you have your B cells, you have in black the antibodies you produced on your own, you have in green all of the antibodies I just gave you through an infusion. The lining of the blood vessel, it takes in antibodies as a part of the normal conditioning of blood, of its support of the blood, and it sorts antibodies between stuff that needs to break down and stuff that needs to get spit out. And you can see here, if you've got 6 of the donated and just 1 of yours, then most likely what's going to get spit out is donated. The key here is you are producing antibodies that damage your own system, your own nerve muscle junction. The donors are not. So if you have lots of antibodies from donors and very few that you made on your own, you're less likely to damage your own nerve muscle junction. Okay. Next question is, does IVIG manufacturer really matter? There are many manufacturers, though none of them are FDA approved for myasthenia, and to my knowledge, none of the manufacturers are currently running trials. The major difference in manufacturers is the concentration of the drug and the liquid the drug is made in. So what is that liquid? That liquid is a solvent. A solvent is what the antibodies are diluted into. And there are some solvents that are not logical to use. For example, a diabetic really probably should not get dextrose. For most patients, this is very personal, and for most patients, this the choice of manufacturer doesn't really make a difference in whether or not their disease is going to respond. Okay. Last class of drugs is the complement inhibitors. So what are the complement inhibitors? These are drugs that restrict your ability of antibodies to actually injure the muscle. So an antibody will attach to muscle, and because it attaches, it will attract inflammation. Complement inhibitors restrict the ability to attract inflammation. So this is a lot like taking a decongestant for your runny nose. You might have a viral infection in your nose. Your nose is responding by producing mucus. If you eliminate the mucus, you may not have a terrible side effect of having the virus in the nose. You might not even notice it. And that's the idea with complement inhibitors. We allow the entire immune system to be intact. We allow your body to produce antibodies. We allow those antibodies to attach to muscle, but you eliminate the ability to then produce inflammation at scale. And when you do that, some patients do really well. They can, if without the inflammation, they can function quite well. For other patients, there are other ways in which the disease is interfering with their muscles, and they're really, they need something more. So again, what does this look like? This is our diagram again. You've got your T cells, which regulate. You got your B cells, which produce antibodies. You've got the antibodies running in the bloodstream. You got the antibodies attaching. Typically, that would induce inflammation and that inflammation would be damaging, but the complement inhibitors restrict the inflammation. So what are the complement inhibitors currently on market? There is one. It is eculizumab. Trade name is Solaris. This is FDA approved. It is manufactured by Alexion, and Solaris is infused every 2 weeks. There is a clinical trial ongoing right now with the longer acting ravulizumab. So ravulizumab is also made by Alexion. It is FDA approved for other diseases. It is not FDA approved for myasthenia gravis. There is a clinical trial ongoing for myasthenia gravis, and this is an every 8-week infusion rather than an every 2-week. The other major company in the myasthenia gravis marketplace right now is Ra Pharmaceuticals UCB. So Ra Pharmaceuticals makes zilucoplan. Zilucoplan is a daily complement inhibitor. It's a daily injection right underneath your skin. And the device that they use for injecting underneath your skin is a very similar device that is used in many other medications that are injected under the skin. So the device itself is really well accepted as having a small needle and this sort of stuff. What the experiments are is whether or not you can dose this kind of drug under the skin and get the same effect. So again, to run through these parts of the immune system and the way they're being targeted, you got T cells, which are really your master switches. And currently we are using some drugs off-label, things like azathioprine or Imuran, cyclosporin, mycophenolate or Cellcept. There is some data to support why we use these drugs, though they are not FDA approved for this disease. And then there's a newer class of drugs on the way, which may help to restore the way that these T cells actually should behave. You've got the B cells. These produce antibodies. These are the factories, and they put antibodies in the bloodstream. You can eliminate these B cells or plasma cells, that is the on-market but not FDA-approved Rituxan or the experimental Takeda drug. But when you do that, you eliminate the factories. It takes months to recover. And so this right here is a diagram of what happens to your antibody levels, and they stay down for months. You eliminate these. Now, you've got plasma exchange, not FDA approved. Plasma exchange, you draw the antibodies out of the bloodstream and filter them away. You've got a brand new class of drugs. This is the FcRn inhibitors. These guys, the lining of the blood vessel is involved in maintenance of the blood. The lining of the blood vessel takes in the antibodies, and the FcRn inhibitors forces the lining to then destroy them. And then you've got the complement inhibitors. The antibody attaches down here to muscle. It produces inflammation, but the complement inhibitor blocks the inflammation. And so as you can imagine, the right choice for a patient and whether or not a patient is a good choice for a clinical trial really depends on what has been tried, what are your particular risks, and in this moment, what are the risks of COVID that you personally carry? And so manipulating the immune system when you have myasthenia gravis is important because we know that the disease, the second word in the disease is gravis. It's a dangerous disorder that can cause you to be hospitalized and have trouble breathing. But how we manipulate the immune system is often a challenge. It's exciting that we may have many drugs on the horizon that are experimental. It's really going to be an important conversation with each patient as to what's the right avenue. And so summary is what I just said. And with that, I think I may have run a little bit over, but I do thank you for your time, and I'm open to any questions.
Thank you, Dr. Sachdev. Nothing has come through chat so far, but I had muted everybody. Sorry, my dog is shaking over there. I had muted everyone when they came on, but you should be able to unmute yourself if you do have any questions for Dr. Sachdev, if you want to go ahead and bring those forward for us.
And I do want to apologize. I had quite a bit of content to get through, and so I did have to do that a bit faster than I typically would. And so it could be understandable if you might have questions later. If you do, I'm sure Alicia can share on the webpage or maybe in the caption to this how you might be able to contact me. Alicia, that would be my work email.
Oh, absolutely. And of course, anyone is always welcome to contact us at MGMI, our website, mg-mi.org. There's a contact us form that you're welcome to fill out. If you received the email today, which I'm assuming everyone on here did, you can also just simply reply to that email with any questions that you have. We're also hopeful to have an event sometime in the month of November where we have our various presenters from our entire annual conference event, and some of our sponsors join us there. So if anyone thinks of any questions between now and then, we'll also be able to have that opportunity in November to go over any of those things that people are curious about. And then, of course, you can always email our office, info@mg-mi.org. I did have one question for you, Dr. Sachdev. If somebody wanted to participate in a clinical trial, where should they start to go about learning more about potentially joining something like that?
So the most important question when someone is interested in a clinical trial is, what has been tried thus far? You know, an experimental medicine might be a good option, but oftentimes, especially with this double-blind phase that you have to go through where you might be getting placebo, if you're thinking about a clinical trial, it's probably that your disease is not as well controlled as you would like it to be. If in particular that issue is something like breathing, endurance, swallowing, stuff that could lead to hospitalization, we're going to want to talk about how your disease is managed now. And so there are some patients who might come to me for their first visit and say, you know, I want to be in a trial. And that's rarely, it does happen, but very rarely is that the right answer first. Clinical trials are experiments, and there's that placebo phase. And so you're going to want to work first with your trusted neurologist, your trusted provider. If then it comes to a conclusion that maybe a trial is the right answer, then you would reach out to our group. We have a research unit, but I would call this clinic number and declare your interest, that you have myasthenia and you want to hear about trials, and we'll reach back out to you.
Thank you. I appreciate that, and that's great information to have for the patients who are listening to the talk today. Did anyone else have any questions for Dr. Sachdev at all about research updates for MG? Well, it doesn't look like we have anyone unmuting themselves, Dr. Sachdev, so we can go ahead, I guess, and we'll wrap this up today. I do want to thank everyone again for joining us on day 9 of our 9-day educational conference event that certainly looks different than they have in the past, but we're very grateful that we were able to offer this content virtually. We also are offering this in a couple of different formats to people living throughout the state who don't have easy access to the internet. So particularly if anyone tuning in, not just on the live call, but anyone watching this later on YouTube or on our event page, if you're able and feel compelled, your donation to MGMI will help ensure that we can continue to offer this important information both online and to our MG family who don't necessarily have easy internet access. So you can always donate through our website, mg-mi.org, or you can text MGMI to 41444, or head to our event page that is linked in the various emails that we've sent, and we'll continue to share the link to that too in any follow-up emails that we have. And we look forward to connecting with everyone soon as we continue moving toward a world without MG. Thank you, Dr. Sachdev.
Thank you.